I want to have a look at preventing disease and perhaps even how to try and treat disease. And with the current pandemic, this probably is sort of quite relevant. So, what we've got in the community, I suppose, is something always called natural immunity. Doesn't matter what sort of disease we have, we often find that there are a few people who are basically immune to this sort of bug that comes along. We've got two sorts of diseases we have to remember. We've got non-communicable and non-communicable diseases simply cannot be passed from one person to another. These are your things like your heart disease. And for many people, these are inherited. And we've got many sort of things like cancer here, many diseases of the nervous system, the endocrine system, digestive systems. All of these are non-communicable, and natural immunity doesn't really play a large part in this. But when we come to the communicable diseases, then basically you come into contact with some sort of foreign antigen of some sort, and you need to have some sort of immunity to prevent you getting the disease. And some of them occur just naturally. So you've got some natural disease, and you may sort of perhaps you may already have some sort of antibodies to this. Now, how have you managed to get some of these things? Well, usually this can be passed on from a parent, typically the mother, via breastfeeding, or perhaps it was across the placenta. So we've got this, and this is called our natural active immunity. So we have this. Now, the immune system of a newborn baby is certainly not mature. It can't make antibodies for the first couple of months. So this is a problem, that we've got a newborn, they just literally cannot make antibodies for the first 2 months. Now, this means that they've got to get these antibodies from somewhere, and this is going to go across something like the placenta, and this is basically the mother simply supplying these things. And when you get the first milk, this is called colostrum, and this is typically very high in antibodies. And this way the mother is transferring as much of her immune system across to the baby as possible. The infant gut allows these what are glycoproteins to pass into the bloodstream without being digested. So within a few days of birth, a breastfed baby will have the same level of antibody protection against disease as the mother. And this is our natural passive immunity. So we've got somewhere some sort of natural active system where you're meeting something from, if we want to put it down, I suppose here it can be passed on from the parent, but you meet for basically the second time, and you've got this natural active immunity, whereas the other one is more passive. It's passed just from the mother, and this is only going to last until the baby makes its own antibodies. The good news is the antibodies the baby receives from the mother are probably relative to the pathogens in its environment, where the mother probably acquired them. So this is our idea of natural immunity. Coming with this, we have artificial immunity. Basically, some diseases kill people before their immune system makes the antibodies they need. And this is a big problem. We are seeing a lot at the moment of the COVID-19 here, and basically it kills before the immune system, I suppose, is ready. It's got things to sort of fight it with, and all we can do for these people is to try and give them some sort of antibodies. So we are seeing at the moment basically antibodies in someone that has recovered. These are collected and they can basically pass these on to the person that's ill. And this is being tried as a way of trying to help things go along. So artificial immunity, we've got artificial passive immunity then. So certain potentially fatal diseases, antibodies are formed in an individual. They can be then extracted and injected into the bloodstream of another individual. So we might take something like a mouse. We are getting that to create antibodies. These are then extracted and once they're extracted, then these are then injected into the individual. And this is our artificial passive immunity. It's basically very temporary, but it might be enough to get somebody over the problem that they're going to die before their immune system's ready, and this might sort of give them the chance. There is another case of something here, which is tetanus. Now, tetanus is basically caused by a toxin. It's produced by a bug, which is Clostridium. Tetani. And the Clostridiums are particularly nasty sort of bacteria. These are found in soil, animal feces, and this causes basically the muscles to spasm, and so you can't basically, you cannot swallow, can't spell, and typically you can't breathe, and this tends to kill people. So what we can do is we can try to extract these from something else, and some animals you can try and get them from people who might be infected with tetanus after a cart or something. They're particularly given this sort of antibody here, which is often extracted from the blood of horses. And that has got the antibodies, or to be more precise, an antitoxin, which is effectively an antibody, as we did last time, and these are injected into someone and they can try and save them. Where else we can try and go is we can have a look at something like artificial active immunity, and this one we know is vaccination. So let's do this one. So we've got artificial active immunity. Now, how do we get this to work? Well, many people are now getting to be quite good at trying to do this. So what we do is, number one, we somehow need to make this pathogen safe. We can try and do this in various ways. We might kill the bacteria, and I'll just say inactivate the bacteria somehow, so that there are basically no risk of getting the infections. An example of a killed or inactive bacteria or virus here might be for something like whooping cough. We can try something else, I suppose, which is attenuated. So, and by that we mean a weakened strain of a particular virus, and this one is rubella. the BCG against tuberculosis, polio. I remember taking polio vaccine orally on a sugar knob when I was sort of quite young. We can have toxin molecules that have been modified, and if these have been modified, then basically they're going to be a little bit safer, and this is a case of diphtheria or tetanus. We can isolate the antigens. And this is what we're doing with influenza and, I suppose, the coronavirus for COVID-19. It's what's largely been used. We can try to make genetically sort of engineered antigens, and this is fast becoming a sort of a growing industry here, and this would be hepatitis. So first of all, we make the pathogen safe, and there are various ways we do that. Next, once it's safe, we can put in a small amount is basically of this safe antigen is injected into the blood. And what this is going to do is going to give us a whopping great primary immune response. Now, if you'd had the disease, this needs to act very quickly. But in fact, here, because you've actually got something that's live, attenuated, dead, modified, somehow, some around here, then it can take a longer time to work, and that doesn't matter. And this basically means that you gradually produce your own antibodies, and you produce things like the memory cells. So you're now able to recognize this, so that when you come into contact, then you get a secondary immune response. And you can therefore start to produce this pathogen destroying thing very quickly. Artificial immunity provided by vaccines lasts an interesting amount of time, and this is one of our problems with COVID at the moment, that we don't know. So these artificial things, these artificial immunity systems, the vaccines here, basically may last a few months, a few years, or a lifetime. And if it's these two, then we need things like boosters. And the other problem that we get with something like flu is that flu changes. So a virus like flu, and we also know this to be true of COVID-19, the coronavirus, they change often. And sometimes they change radically. If they change often, then it's a case of basically modifying the vaccine. If they change radically, then you need to start from scratch. So with normal sort of influenza, we can adapt it. But when it suddenly changes, and that's what we were expecting for this epidemic, which became a pandemic, we thought it was going to be flu doing that because flu kills, and we've got the story about the Spanish flu in sort of 1920, that sort of time, and that killed an enormous amount of people. And because we'd seen that, we were hoping to be prepared. But the answer is no, we weren't. We were prepared for this, but when something else comes along like the coronavirus, we just weren't prepared for it. And it takes a long time to start to sort of understand how it works. We see quite a lot of complaints about governments that they did this wrong, they did that wrong. But I think generally that's rather unfair on the governments and the scientists, because when these diseases came along, we didn't know. We didn't know what they were going to be capable of, what they were going to do, exactly how they were transmitted, what the follow-up was going to be. And so we had to go with the best advice we could. And people complain because this best advice changes. And it does as we learn more and more about these different sort of viruses and what they do and how they work. So influenza was one of our favourite sort of things to look at. And I suppose we better define a couple of terms. So let's try and do that. And the first of the terms we reach is an epidemic. Right, so what is an epidemic? Well, an epidemic is simply something that's going to go through a lot of people in a single country, usually. And it spreads across this country. Whereas our pandemic, the same disease now goes across many countries, or in fact continents. And this has got worse because of things like better communication, mass transport. So better communication, I mean by the fact that someone can be on a plane from one side of the world to the other and spread within 24 hours. And sometimes that might be before people even are aware they've got the disease. At the beginning of an epidemic, then to try and cure this, or in fact a pandemic, what we need is mass vaccination. And this works. However, of course, if we don't have a vaccine, then as we've seen with COVID-19 spreading, there's very little that we can actually do to try and sort of fix it. The case studies we normally look at in this are looking at influenza, and basically we look at these sort of things because we know flu, basically we get new strains formed, and these are simply formed basically by mutation. Mutation also occurs more often because of the speed of the life cycle. The faster the speed of the life cycle, the greater the amount of mutation opportunities there are. And the problem is with flu that we can get some sort of crossing of the species barrier. And of course what I'm saying here for flu also is true of the coronavirus, where we actually met a novel new coronavirus which basically crossed the species barrier. So although people may be and animals may be immune to this particular disease, as soon as it crosses the species barrier, it brings with it new antigens. And that means that people are susceptible to it, so these will basically not be recognized. And if they're not recognized, then we've got a real problem. Every year in the UK, older people and anyone who's got a compromised immune system are given flu vaccines, and that's done to try and cut down this flu epidemic from sort of coming on. Basically, if a flu epidemic begins, more people are vaccinated to control the infection rates. But because people travel freely, then it can spread from one part of the country to another. And that's why we needed ways of trying to stop this, and effectively the only way to do this is the lockdown. If we can restrict contact, then we can try and stop these diseases from spreading. But we do have a problem, and that is, how do they spread? We've got a problem with how they spread. The easiest one to cope with is touch, and if you'd have basically zero contact with people, then it's going to work. We've got things that are waterborne, and basically this is usually, but not exclusively, bodily fluids. And simply by having zero contact, we can again hit that. Our real problems come when something is airborne. And as soon as something becomes airborne, we've got a problem. And we're looking at a figure of here 2 meters for COVID-19. But some of the latest research is saying that that is at, in fact, 4.5 meters. And what governments are doing is they're lowering this down to 1 meter. They want to, because what they want to do is they need to get the economy recovering. Yet maybe we ought to be thinking about making things get further apart. One way recovers the economy, but the other way means that we can enter the second wave. And the second waves typically are much worse than the first. If we have a look at the flu, and this was sort of our 1918, 1920 sort of going on here. Then this sort of hit a small peak and then a very much larger peak. And here we saw probably, I think it's about 10 million died. The statistics aren't so good on this because it didn't record exactly how people died. But here, this is our first wave, and typically we have a much larger second wave. But whether all countries are going to manage this, I worry about places like America, United States especially, but also countries like Mexico and a few others. They have a very, very large first wave. And if that's a very large first wave, are they going to perhaps have an even larger second wave? And the only way to stop this is with a vaccine. And at present, of course, we have no vaccine. Big problem. And until we can get a vaccine, perhaps things around the whole world won't return to normal. So what we've got to try and deal with are various things like medicines. So if we can't stop it, so you can stop or you can prevent is a better word with a vaccine. If that option is taken away from us, then we have to look for some sort of cure, and this needs to be something like a medicine. For bacteria, we've got an old ally, and this is the penicillins. The first widely used, effective, safe antibiotic. Comes from the mold of the penicillin, right? Discovered by Alexander Fleming in 1928 when he made a sort of an accident, really. But it needed a couple of guys, Howard Florey and Ernst Chain, to basically develop an industrial process for producing this drug. And now we can use enormous sort of resources here to sort of model drugs, to work out three-dimensional models of what's actually going on, to try and sort out this system. But we've got basically no new antibiotics. nearly all of them are based on penicillin and the way that it works. So we've got to try and find perhaps new ways of trying to deal with some of these. Most of these drugs, these medicines, come from living organisms. Other living organisms are fighting these things, and some of them come up with a good sort of way of trying to sort of get rid of them. We've got various things to try and get. So we've got penicillin. This comes from a mold, and a mold growing on melons. Now we've got other ones which are quite interesting. You've got a chemical called docetaxel, which is from yew trees, and this is an anti-cancer drug. My favourite, aspirin, which comes from the willow tree, and basically this is a very simple painkiller, but it's also quite an effective heart medicine. Prialt comes from the venom of a snail. And this is a fantastic painkiller. And from the evidence, it's probably 1,000 times better than something like morphine. We've got another of these sort of antibiotics. Vancomycin here, and this again is from a fungus, and this is perhaps our most powerful antibiotic. If you haven't heard of it, it's because we save this one up until everything else fails. And then there's digitoxin, which is based on digitoxin from foxgloves, and this is a heart drug. A little bit kills you, but a very, very small amount can save someone's life. We're also looking here then at an idea of drug design, and we're getting better at trying to design our own drugs, and we're looking at something like personalized medicines, that you get the specific drug that will work for you. and so we can modify the drugs, or we can give you a combination of different drugs that will work best for you. And this is sort of a real growing market when we can get there. Related to this, I suppose, is synthetic biology. Synthetic biology, another step here, is basically using lots of genetic engineering to sort of try and beat the things. We can re-engineer biological systems, we can create new drugs, and we can create new ways to deliver drugs. But we're stuck when we come round everywhere to our antibiotic dilemma. And this causes us really a lot of problems. If we go back to the beginning of the 20th century, thank you, that was no help. Talk to the computer, it doesn't sort of take any notice of you. Beginning of the 20th century, 36% of all deaths came from communicable diseases. That is a lot, and enormous number of children died because of this. Now, over the years, this has dropped dramatically, but what we're now seeing is that antibiotics are becoming less effective against the treatment of bacterial diseases. Bacteria are becoming more resistant to more and more antibiotics. First it started with penicillin, and now there are some microorganisms that are resistant to all antibiotics that we have. And this can mean that perhaps when we get into the 21st century, we are looking at 36% again of deaths caused by communicable diseases. We got it right down, and this is a real worry. We've got things like MRSA, methicillin resistant Staphylococcus aureus, and there's another one, MRCD, methicillin resistant Clostridium difficile. Basically nasty little bugs, and we've got big problems with these. MRSA is basically carried by about 30% of the population in the last study that I was having a look at. Right? In the body it causes boils, abscesses, septicemia. We've also got here the other one, which is Clostridium now this lives in the gut of probably about 5% of the population. It produces toxins in the intestines, leading to diarrhea, bleeding, possibly even death. It's not a problem for a healthy person. They get a bit of diarrhea, they survive. But if you're ill, this one helps you to die. So, how do we solve the problem? Well, solving the problem is really down to you. Trying to work out how we can make new antibiotics. Trying to work out what we can do to try and fix some of these basic problems is now going to be down to you. Can you find more different ways of killing bacteria, more ways of killing viruses? Can you find more animals? And we're shrinking the gene pool, which is a problem. So that's a big problem that we've got, and it's one that I can't answer at the moment. We are working on designer drugs, we are working on vaccines, but as you see with COVID-19, it takes a very long time to develop one of these things. People get impatient. They say, I want a vaccine now. But it takes time. It might take a year or two to develop a vaccine, and by that time we're looking at things like COVID-19 causing lots of problems. We do find that some things seem to be working. We find that children don't seem to suffer from COVID-19. Why not? Could it be linked to the fact that they've had perhaps some other sort of vaccination, and it might be also preventing or making it sort of less likely for the coronavirus to sort of cause so much problems for them, so they become just carriers of this disease rather than sort of passing it on and dying themselves. They just pass it on. Big problems, and those are for you to try and solve. If you found this useful, then please subscribe, give us the thumbs up, it's always nice, and I will see you next week when we do some more A-level biology, topic by topic. Until then, stay safe. Bye bye.